All right, thanks, Dr. Palmer. And let's move on now to institutional governing policies. Dr. Zach Adelman from Virginia Tech University.
Okay, everybody's made it so far, so I'll be the last one today. And I want to thank the committee again for having me back again. I'm going to take off the Gene Drive hat that I wore last week during the webinar, and I'll move towards the Institutional Biosafety Committee chair position that I've been in for the last 4 years and tell you about how our committee at Virginia Tech and as a representative of IBC might view gene drive technology in the context of the other things that we look at. So this paper has come up a number of times in the various talks today, and I'm just going to go over this again in a little bit more detail, talking about laboratory gene drive experiments. But when you actually read this paper, the title should be more along the lines of how to prevent an accidental gene drive. It's kind of a guide for people who say, if you don't actually want to do this, here's how you can avoid making this by accident, because it's so easy to do. But it doesn't really seem to apply to people who actually want to make a gene drive system or a genetic element capable of gene drive, to be more precise in my language, for those groups. For example, if you're going to separate the components, then that's not actually what you're going to test in your field trial. So all of your data is not really going to apply, because to go to a field trial, you need the data, the regulators are going to need the data you have in the lab first. So you can't separate it and put it back together later. You have to actually test the real system. Just like you can't test a system where they can't reproduce in the wild, because what are you making it for if you can't do that? So this is great if you don't actually want to construct a gene drive system, but if you actually do, you can't do those two things. And then the third one, ecological, you can't really do that either, because this is the one that really got under my skin. All right, you can look at the text. This is a laser pointer, right? You're allowed to work on gene drive systems in Anopheles in Harvard or Southern California, but you're not allowed to work on them in Africa. Okay? And that's what they're saying in this paper. And of course, that's not really what we want to be promoting either. We want people to have capacity building in their native countries. We want people to be able to use these technologies responsibly in their own countries to solve their own problems. So that doesn't really leave us with any of those other three options. As far as containing these experiments in the laboratory, we're left with where we started from, which is the physical barriers, the physical containment, and I'll add in my own box right below that, which is training and work practices and emergency planning and all these kinds of things that take the human element everybody's been talking about, or not everybody, but we've heard a lot of talk about, well, human error is there, so you can't rely on containment. Well, it's like human error is there in pretty much everything we do, but we still manage to go to the moon, go to Pluto, go to all kinds of places through planning, through saying what are the things that could go wrong, what happens if we, what do we do in that case, and think about everything you can think of, and you'll do okay. So to continue on, piling on a little bit, some other phrases that I kind of picked out. All work should be preceded by assessment by relevant biosafety authorities, but those were never called out by name as to who that might be. And then we can't ship or use these things until we have formal biosafety guidelines, which I'm going to argue that we actually have those. And these have been mentioned various times. These are the NIH guidelines for recombinant DNA, which are also not called out by name in this paper. So as has been mentioned, we've had guidelines for how you use recombinant DNA in the laboratory environment since 1976, last revised in 2013. Again, any type of inserting synthetic DNA, recombinant DNA into the genome of an organism, you're going to fall under these guidelines to some extent. Okay. And what's important to note is, though, even though gene drive, the phrase gene drive or the concept of a gene drive system is not specifically called out in these guidelines, NIH specifically wants you to look at the intent of the guidelines, how they were designed, and what the purpose is of having those there, and use your own judgment as an institution with their guidance to kind of regulate these in turn. And so certainly all these gene drive type systems, all these genetic elements that are capable of gene drive are going to fall under these systems. Okay. But here's where the biggest misconception is that I run into as an IBC chair. Okay. Guidelines. It's not guidelines. And this is one of the recommendations I would love to see. Ask NIH, please, to call these rules, not guidelines, because to drive the point home for various PIs. Okay? If institutions do not follow these guidelines, okay, they can lose their NIH funding for the PI whose work is involved in, or all the NIH funding they get, period. Okay? NIH can just take that away if they don't follow the rules. So institutions have a very strong incentive, much more so than a single PI who might be upset that their research is being delayed okay, because they have a $1 million grant or a $100,000 grant. You're talking about hundreds of $1 million in NIH funding that an institution might lose if they cut corners on these guidelines. So they are strongly incentivized to follow these rules. Okay, so to talk more specifically about how these rules factor in to various genetic systems that might be capable of gene drive, NIH has this kind of hierarchy of who looks at it and when and at what point. And so we can start out at the lowest part of the tower. These are experiments that have been considered exempt from the NIH guidelines. Yes, they involve recombinant DNA, but NIH has deemed that they are so harmless and so safe that we don't actually have to review them at the level of the IBC. And so if you were going to generate a system capable of gene drive in Saccharomyces cerevisiae or common baker's yeast, this would be exempt as of right now from the NIH guidelines. The IBC at that university would never see it. Okay? If you were going to generate a system capable of gene drive in rodents or in most crop plants, that would go under Section 3E, okay? And that means that the IBC can be notified when the PI initiates that work, but the review actually would take place sometime after that, probably within a month or two of that work. So the work would initiate, okay, on that gene drive genetic system before IBC has really gone over all the risks, all the procedures, and everything like that. But once we start getting into animals, and this is going to include everything that's not one of these things listed here, now you have IBC approval required before project initiation. Okay, so that means the IBC has to look at everything before you start anything. But that's not the top of the tower. Some experiments have been deemed that the local IBC approval is not sufficient. Right? There's an additional layer of oversight. And this is, again, one of the things that the committee can look at is do gene drive, because all of this, right now, the gene drives are just fitting, or I say gene drives, but genetic elements that are capable of this gene drive are not treated any differently than any other genetic element that is inherited normally in terms of how it's looked at in the NIH guidelines. Okay, but that can be changed. Above that, you have IBC approval and also recombinant DNA advisory committee. Okay, and this is something at a national level. And so human gene therapy experiments, okay, have an additional layer of scrutiny on top of IBC approval. Okay, you can go up even higher if you're going to clone some of these potent biological toxins into an organism and have those be expressed. Now you have the Office of Biotechnology Activities at NIH who is going to review that work in addition to the IBC. Okay, and at the top of the tower, where the director of NIH, OBA, and RAC will all look at, is this generating microorganisms that are resistant to different molecules. So this could be antibiotic resistance in a bacteria, it could be an antiviral therapy that's put into a virus, where we would then compromise the ability to treat that infection in the real world. So there are levels of risk. There's already a pathway and a pipeline for how to conduct higher levels of review when the feeling is that there's a higher level of risk. And so simply by moving some of these gene drive experiments up the tower, you'll gain more oversight. And the pipelines, nothing has to change, it just moves up the tower. Okay. And again, I want to reemphasize for the work done that's been described in Drosophila and anything that would be done in these mosquito species, the local IBCs at any institution, and this could be academic, institutions like we have at a university, but it could be a medical school, it could be any institution that receives any NIH money has to follow these rules, okay, not guidelines. But all this work has to be approved by the IBC before it initiates. Okay, actually, any transgenic Drosophila you make or get from a transgenesis facility has to be approved by your IBC before you work with that. And a lot of people may or may not know that, and that might have been part of the impetus behind that publication, is that that may not have been known. But this work has to be reviewed on any transgenic before it proceeds, especially for these kinds of things. The NIH guidelines don't just spell out what needs to be reviewed, they spell out very clearly a standard of containment that should be maintained to give the IBCs some guidance in how these kinds of experiments can be conducted safely or how risk can be managed for a lot of these things. And we have guidelines in Expandix G. You can look at containment in the laboratory environment, and it can go from BSL-1, and then BSL-2 builds around that, BSL-3 builds around that, and BSL-4 builds around that as a hierarchy, again, of containment. But some things don't fit into the laboratory, so if you wanted to work on transgenic trees, now you're in greenhouse-based containment, but there are guidelines spelled out. How do you work with a tree that's maybe in conjunction with a dangerous plant pathogen that's recombinant? Okay, you can go all the way out to greenhouse level 4 And there's guidelines for how to do that. The same thing with large animal in these guidelines here. So there's a lot of guidance that's given to local IBCs to use their discretion as far as how to conduct this work under rising levels of containment based on the risks that are presented and identified. Okay. So the NIH guidelines also spell out who's responsible for what in a very clear and unambiguous manner. What is the institution responsible for? What is the IBC responsible for? The biological safety officer and the PI. Okay? And this is, of course, very helpful in all these things. And one of the things the institution is responsible for, of course, is forming an IBC and giving it a charge and giving it the freedom to operate. And one of those freedoms is to be able to allow or deny any work done on campus. Okay? And the IBC has that authority. And we have, on rare occasion, but on a consistent occasion, we have denied PIs the ability to do certain lines of research for various reasons, but we have exercised that authority. Most of the time we'll work with a PI to try to find some kind of common ground where the work can be done safely, but in some cases it's just not possible and the work is just not allowed. The IBC is also responsible for reporting. If there are incidents, if there are accidents, if there are any kind of releases, injuries, anything, that has to be reported back to NIH, and those statistics are accumulated, and then we work out different procedures. What can be done differently? What can we learn from this? How do we improve our safety? And of course, NIH is then auditing all that material and then giving advice back to us as far as what we can do. Who's on your IBC? NIH actually wants to know. It can't just be anybody. They want the list, the biosketches, because as we've heard, if you don't have the right expertise reviewing this material, then you're going to miss certain details that are really important. Okay, so our committee contains expertise on people who are working on recombinant bacteria, recombinant protozoan parasites, recombinant viruses, recombinant animals, large animals, small animals, recombinant plants. The whole gamut. We have public health experts, epidemiologists, veterinarians, and of course we have community members as we're required to have. Okay? And the NIH looks at that every year and says, is this a valid IBC? Do they have the expertise necessary to evaluate this work or not? And if they don't accept our roster, then our work is not legitimate and our institution is now not in compliance. So for those that have been trained more recently, IBCs are kind of something that you're more familiar with, but that's because they've been increasing in number in recent times. But if you're trained kind of a little bit farther back, you may not be as familiar. I marked these two arrows. This is where NIH announced the doubling of the budget back in 1998, and you can see the trajectory of the number of IBCs that have to deal with recombinant DNA starts to increase at that point. The slope changes again around 2001, when all of a sudden we have a large influx of biosafety research, a large building boom in BSL-3 research, and now we have a lot more work done in recombinant pathogens that have to be held at various high containment labs. And that trend has continued, and we've gone from 20 or 30 years ago from just 200 IBCs to almost 900. So what does the IBC actually review? And it's exactly what we're talking about when we talk about genetic elements capable of gene drive. We're looking at the risk to human health. So do these recombinant or naturally occurring organisms have any kind of implications for the health of the workers, the researchers that are handling these, and the risk to the environment? What happens if these get out? Can these pathogens or organisms establish themselves in the wild? Can they interbreed with wild things? Whether they have any kind of ecological effects in the area? These are the questions that the IBC is set up to investigate for any transgenic, for any recombinant DNA-containing organism. And so this concept of a transgenic gene cassette that's capable of gene drive, they're going to be looking at the exact same pieces of information. And the Virginia Tech IBC, every IBC is going to regulate plus or minus more things. We regulate a lot more than just recombinant DNA-containing organisms. Anything that's infectious at a biosafety level of 2 or above, we are going to regulate. We don't regulate level 1 organisms, but other kinds of toxins. We do human cell lines, human tissues, things like that, select agents, prions. We are actually the dual-use research entity, so the institutional review entity. We identify dual-use research in the protocols that we review because we do see everything of an infectious nature, and then, of course, synthetic biology. And every IBC will regulate plus or minus more of those. So what are we looking at? Okay, here's this word risk again, and risk assessment, basically trying to say what is being used? What is everything you've got lay out on the table? So even if you're only doing recombinant DNA inserted into this one organism, we want to know everything that you've got in your lab, every living organism, every pathogen, every microorganism, whether or not they're pathogenic, and how these things might interact with each other, and what else is going on there. So what are the agents, hazard materials, what are the procedures, what's the expertise of the PI that's going on here? And I have to say that most of the protocols that we end up denying are due to a PI who does not have any expertise or has insufficient expertise with a pathogen system or a transgenic system that they're proposing to work with. And those are the ones that frighten us the most. And then what kind of surveillance or monitoring is required to do that work safely. Okay. We review, our documents end up for each protocol that we review. This is probably 150 to 200 pages worth of documentation that we ask for from PIs. Reviewing work practices, SOPs, their biosafety manuals. We inspect the labs, look at the facility, look at their security, training documents that they use, pretty much everything that we can get our hands on to assess the scope of the work. Okay, and then that, again, is monitored post-approval through periodic inspections, and we work back and forth. This is a very iterative process. We heard that last week that the group in San Diego was very iterative with their biosafety team, and this is what we do with every protocol we've got. It's a very back and forth process. Okay, and this is a question that actually came up at the webinar last week, and listening in, I wasn't satisfied with the answer, so I kind of incorporated this in. PI suggests the containment conditions and writes SOPs that the IBC considers. The PI leaves the room, and NIH is very specific that the PI cannot be involved in the review of their own protocol. So it's open to the public, so everybody in the universe can come to that review and watch and listen, but the PI and anybody involved in the research, anybody who has a conflict with that research, cannot be involved in the discussion. The IBC then discusses and sets the containment, and that is it. That is what has to be used with that either agent, pathogen, organism. Those are then their binding rules, and that cannot be changed. The PI is not happy with that. They can petition, they can make an amendment, they can offer other suggestions, but the IBC has to consider those too. So at the local environment, the IBC sets the rules on behalf of NIH, and our charge comes from NIH, who has charged the institution. The institution passes that charge to the IBC, and the IBC sets the rules for the PI, and the PI is not on their own. They have to listen to that, or the work will be shut down. And literally, there is one case where we had to go and send somebody and lock the door to a lab and lock the PI out at one time. Okay. But what happens if the IBC is not sure? We have a new technology, different technologies come up now and then. We go to NIH, we go to OBA. They're very, very responsive. I go back and forth with them. If issues come up once every couple months, I send them an email. They get back to me right away. They dig into different matters. If there's an agent we're not familiar with, a practice we're not familiar with, they provide a lot of expertise to IBCs in the cases that the IBC does not have the local expertise to handle that. They also issue guidance documents on specific topics that might be of interest to a lot of different IBCs or things that other groups may or not be very familiar with. Here's two documents on cloning of risk group 4 viruses, or guidance on research with lentiviral vectors, okay? And putting out a guidance document on processes that are capable of initiating gene drive might be something that we do. I'm sure NIH would be very happy to get involved in that. Okay. PI's responsibility after the IBC has approved something like this, again, make all their protocols and manuals available to all personnel. They're responsible for training their personnel and documenting that training has taken place. They have to maintain those written records. We send a biosafety officer and environmental health inspectors, and they inspect all that training documentation on a regular basis. They make sure that it's there, and if they're found not in compliant, we make sure that there's remediation for that. And if there are any accidents, then we have to know about that immediately, and we correct those errors. Okay, so here's the concept. Is there a precedent for these type of gene drive scenarios in the world of biocontainment? IBCs may not have seen this kind of technology before, where you have this kind of biased inheritance of a gene, and of course I would agree with Austin's definition that anything that can be considered a gene drive is when you have biased inheritance like this, okay? But realistically, this is not any different from a scenario where you have an organism infected with a pathogen that's only transmitted vertically, not horizontally. So if they're able to pass an infectious agent onto their progeny, most of those progeny will end up being infected. And functionally, that's not much different than something being incorporated into a genome or being a free-living organism in the germline of that. But we can treat this as a biocontainment issue, treat this as if, at the very basic level, these selfish genetic elements like a transposon or homing a nuclease, anything that's capable of propagating itself or biasing inheritance in favor of itself, is truly the most primitive infectious organism. These are infectious agents. Okay, they infect the germlines and they propagate through germlines, but that's how they complete their life cycle, if you want to call it that. So calling these infectious agents and thinking them as infectious agents rather than strictly genes kind of clarifies things a lot, because IBCs are going to have a lot of experience handling the containment of infectious organisms. And of course, these are going to be things that only infect vertically and only infect the parent organism and can't be transmitted to the worker or the laboratory research, are going to be a lot safer than some, if you have genetically modified mycobacterium, if you have influenza that you're reassembling from various pieces, or plasmodium parasites that have been genetically modified. If we can contain these things and work with them safely, maybe at level 2 or level 3 or what have you, okay, we can contain larger animals where we have much better control over the reproduction and their numbers. Just like we have safety considerations, we have these different risk groups for different pathogens, things that go from risk group 1 through 4. We can have the same kind of classification, and I don't mean to imply that things like transposable elements should be handled under spacesuits like you would Ebola, but you kind of can establish this hierarchy, because we see this kind of fixation, again, on CRISPR-based gene drives or cut and repair-based gene drive systems, but really this is just one type of gene drive. Okay, and we have these other things like underdominance, where if these escape from the laboratory, they're actually much less fit. Okay, their progeny will be much less fit than just a wild-type organism, whereas other things might be relatively easy to get out of the environment, some things relatively difficult to get out of the environment. But how an IBC might handle those might be based, again, on how much risk it is of being established in the wild, and then, of course, what goes along with that. So try to just wrap up. NIH guidelines regulate most laboratory-based gene drive experiments. IBCs act on behalf of NIH to set containment, okay, not the PI. The PI is actually bound by their local IBC. Now, whether an institution's IBC kind of puts its foot down and establishes that authority and uses it, that may vary from institution, but they certainly have that authority, and it's the institution's responsibility to make sure that that work is done safely. IBCs may not have experience with these self-propagating genetic elements, but they certainly will for infectious agents. Okay. In contrast, PIs proposing these type of experiments may not have any experience working under an elevated containment, and herein lies one of the big problems with awareness amongst PIs, that all of a sudden the work they're doing in an organism that they've worked with for a long time, it now needs to be done in a containment that they've never worked under for reasons that they may or may not agree with. But from an IBC's point of view, now because these genetic elements are more infectious to their progeny, that these elevated containment conditions become necessary. So there's precedent, again, for this in the NIH guidelines, so this would be nothing new. Okay. Under the current guidelines, gene drive experiments involving plants, rodents, or yeast may not be reviewed. But for example, if you have experiments involving a replicon, a viral replicon, so it's able to infect its cell but not replicate, okay, that's going to go under this Section 3E. The IBC will look at it later. You can start working up, the IBC will look at it later. But if you restore that replicon with a helper virus, now the IBC has to look at it ahead of time. Same thing with plants. If you're working with transgenic Arabidopsis plants just by themselves, the IBC will look at that later. It's not really that high risk. But if you're using those same plants with plant pathogens that are plant level 2 or higher, the IBC needs to look at that ahead of time. So the same kind of thing, a gene drive system or a genetic element capable of gene drive may, in these other organisms that are normally used with recombinant DNA, it's not that big a deal. IBC will look at it later. But now these can be considered infectious, so they should be moved to a different category where the IBC will see them ahead of time and can and regulate in that way. Okay, so here's my final thoughts. I disagree that there's a crisis in laboratory management of these type of experiments, with the exceptions of things like rodents and plants, where the IBC may not see those ahead of time. Okay. Specific reference to gene drive type systems in the NIH guidelines or some kind of guidance document certainly will help raise awareness among PIs and amongst IBC members, and that would certainly be helpful. Okay. NIH, and here's the biggest point which we've heard repeatedly, is that this only applies to laboratory research. Okay, this is not going to apply to any kind of semi-field trial, any kind of field trial. We have no real pathway there, and that's, I think, where the big bottleneck is, the big pot of uncertainty is what do you do once you finish in the lab and you have promising data? There's really no place to go from there. And I just want to thank these individuals that I pillaged some of their slides and repurposed some of those for the purpose of this talk, and thank you.